So, hello everyone. I'm Nikki Blunier with the IPM Institute Sustainable Communities Group. Thank you for joining us for today's webinar, From Field to Skin, Exploring Environmental and Personal Protection Interventions for Tick Management, presented by Dr. Danielle Tufts, Assistant Professor in the Department of Infectious Diseases and Microbiology at the University of Pittsburgh School of Public Health. The Tick IPM Working Group is funded through the North Central IPM Center, supported by the USDA's National Institute of Food and Agriculture. The Tick IPM Working Group is part of the IPM Institute, which was founded in 1998 with the goal to increase adoption of IPM in agriculture and communities using an educational and solutions-based approach. The organization is split into two groups, the Sustainable Food Group and Sustainable Communities Group. So Tick IPM Working Group is part of the Sustainable Communities Group, and we focus on integrated tick management and education around ticks and tick-borne diseases. We also host the annual virtual Tick Academy and produce educational resources. We also run the Pest Defense for Healthy Schools website, which has a focus on educating K through 12 staff about IPM and best practices to limit exposure and impacts to students. And there are free courses on our website, pestdefenseforhealthyschools.org. And then we also have a certification program called Green Shield, and it's a certification program with a mission to reduce use of pesticides and provide best use practices for pest management professionals, facilities, and landscape companies. You can follow us on our social media, YouTube, X/Twitter, or Facebook, and these are our social media handles, and we have recordings of previous webinars on our YouTube channel. And I want to welcome Dr. Tufts. Like I said, she's an assistant professor in the Department of Infectious Diseases and Microbiology at the University of Pittsburgh School of Public Health, and her research focuses on vector-borne diseases, host vector pathogen interactions, pathogen transmission pathways, ecological drivers of pathogen emergence, and tick control interventions. Um, I am going to stop sharing my slides and I'm going to uh, launch a pre-question for this webinar um, while Danielle shares her slides. And this question is just to um, gauge audience participation. So we'll ask it at the beginning of the webinar and at the end to see how people's answers have changed. So we won't give the answer right now, but at the end. And you can go ahead and share your slides.
All right, thank you. Make sure that I have this going. There we go. Can you see my slides?
Yes.
So, Nikki, I think questions are saved till the end, but is that right?
Yeah, at the end, we will have people put any questions in the chat or raise their hand.
Okay.
I'm on a Zoom call.
Um, okay, you can go ahead and get started. Thanks.
Okay. All right, thank you everyone for joining, and thank you, Nikki, and the IPM or the Tick IPM working group for inviting me to talk today. So I'm going to be talking about some of our interventions and why aren't they advancing? There we go. So just to kind of give you a roadmap, where I'm going to give you some background information, and then go into some of our studies about the participatory epidemiology, which encompasses survey information, and then our field-based studies for tick control, and then some lab-based studies that we're working on. So as I'm sure most of you guys know, the tick-borne diseases are the most common vector-borne disease in the United States, And Lyme disease is estimated to affect about 500,000 people annually. So you can see that there have been Lyme disease cases across most of the US, with the majority in the East and the upper Midwest. And until recently, Pennsylvania has been the state with the highest cases of Lyme disease for about a decade. So Pennsylvania is this green line here. Um, and so we have Pennsylvania as one of is was the top state until just recently when New York kind of took that over. And Ohio isn't in the top 10 states for Lyme disease cases. And so this is considered an emerging area. And so this leads me into studies on people's perceptions of Lyme disease. And so participatory epidemiology is this approach that kind of integrates community knowledge, experiences, and observations into epidemiological studies to help improve disease surveillance, control efforts, and prevention. And so homeowners are an important demographic uh, due to their potential exposure to ticks and tick-borne diseases while on their own properties. And several studies have investigated perceptions of homeowners and their perceived risks of Lyme disease and other tick-borne diseases using knowledge, attitudes, and practices, or CAP surveys. And so this was a recent study of homeowners from an endemic area of Block Island, Rhode Island, and an emerging area in Staten Island, New York. And they showed that there are significantly more people think that ticks are found in the parks compared to other locations, and fewer participants thought that ticks could be found in their own backyards. Um, Additionally, there were significantly more participants from Block Island that checked for ticks and avoided outdoors compared to Staten Island residents. So these people know that there's ticks present. It seemed like the Block Island endemic area did things differently, significantly more so, did more tick checks on themselves, and also used more protective clothing and things like that compared to ticks more in an emerging area. So this is a map that I made that shows which states have been studied regarding residents' perceptions of ticks and tick-borne diseases. So that's all of the green states where we have survey data. But there's been no data for Pennsylvania specifically. Pennsylvania has been kind of lumped into a northeastern group, but there was no information about the residents themselves, their answers to questions, and that could also be just from the eastern part of the state. So it wasn't clear where in Pennsylvania those responses were coming from. And this is kind of problematic because, as you saw, Pennsylvania is one of the most heavily affected states for Lyme disease cases. So the yellow states are those that are in that top 10 list of states with the highest cases of Lyme disease that we don't have any survey data from. And so since Borrelia burgdorferi, the causative agent of Lyme disease, can affect humans and domestic animals, Lyme disease is an important One Health concern between humans and animals. And so people residing in these endemic areas are likely to possess differences in knowledge and protective behaviors compared to people living in areas where Lyme disease is newly emerging. So to better understand behavioral differences and perspectives of Lyme disease and other tick-borne diseases in these areas, we used elements of this participatory epidemiology by studying homeowners from Lyme disease endemic areas, which is within southwestern Pennsylvania or western Pennsylvania, and an emerging area of eastern Ohio. And so this really helped us to better understand the needs in these areas for control and prevention strategies. So our survey covered three main areas of interest. The first was perceived risk of tick exposure and tick-borne disease health risks, where we asked questions like, do you suspect that there are ticks on your property? And if you find ticks on your property, do you think that they pose, or do you think ticks in general pose a health risk to your family? We also focused on personal protection methods used to prevent or reduce exposure to tick bites. And we asked participants, what personal protections, if anything, that they use to avoid ticks when they're on their own property. So that was a distinction when you're on your property or when you're out in like a park area or green space. And then the third area that we wanted to focus on was protection methods to reduce tick presence on residential properties. And so we asked questions like, have you used any pest prevention products on your property? And if you find that there's ticks on there, would you do anything differently? What actions would you take? And so, as I mentioned, we used two different areas. We used an endemic area for Lyme disease, which is Western Pennsylvania. And we used different methods for recruiting participants where we physically went out and knocked on doors. And if we didn't get in contact with someone, we left door knockers or flyers with information about the study. And we also made an online survey for people to be able to complete. So in Pennsylvania and Ohio, which was our emerging area for Lyme disease, we visited more than 200 homes in several different counties. Um, and we did have more people that were interested in the study, but some of them didn't meet our inclusion criteria. But we had quite a good response from some of these people that did fit our criteria. So for our first questions on residents' perceived exposure and health risks, we found that when people were asked, do you suspect ticks on your property? Um, Significantly more people in Pennsylvania said that yes, they found ticks on people or pets on their property compared to that people that found nothing. And there was no significant difference between the states, but we did see significant differences within states. And so both states reported finding ticks on their properties or people or pets. For our second question, again, we didn't find any significant differences between the states, but homeowners in both states reported that they felt that ticks did pose a major or minor health risk, significantly more than those that thought that they didn't pose any risk at all. For our questions about personal protections people use, Pennsylvania homeowners use some type of personal protection, significantly more compared to Ohio homeowners, where Ohio homeowners were significantly more likely to not use any protection at all compared to Pennsylvania homeowners. And within Pennsylvania, these homeowners used protective clothing, performed tick checks, and showered or changed their clothes after spending time outdoors significantly more often than doing nothing. And they also performed tick checks significantly more than using tick repellent. So while tick checks seem to be the highest form of protection in Ohio, there's actually no activities that were significantly different. Tick checks were, excuse me, were also the most common behavior reported from other endemic areas such as New York, New Jersey, and Wisconsin. For our questions about control efforts on people's properties, we found that significantly more Pennsylvania and Ohio homeowners did not use pest prevention products on their properties compared to those that did. So most people decided not to use any type of protection on their property itself. Um, and Pennsylvania homeowners were significantly more likely to use personal protection than to treat their properties or use a pest management company. In Ohio, homeowners were significantly more likely to do nothing nothing differently than to consult a pest management company. So these findings were in contrast to some other endemic areas, such as New York and New Jersey, where homeowners were more likely to treat their properties with pesticides or use pest control methods. So this was really interesting and kind of helped us when we moved on to our field-based interventions. So some of the takeaways from our survey data is that homeowners in both states reported seeing ticks on their property or on people or pets, and they believed that ticks were a health risk. However, most people weren't willing to treat their properties at all, and they generally opted for personal protection methods such as tick checks or protective clothing. So we also used this survey to recruit homeowners for our field-based tick control interventions. So once we understood what the homeowners thought about the ticks and tick-borne diseases on their properties, then we asked if participants were willing to allow us to evaluate the effectiveness of different tick control products around their homes. So that leads me to our next field-based stuff. And as I'm sure most of you guys know, this is kind of a review for everyone here, but the tick and the pathogen Borrelia burgdorferi life cycles are really closely linked. And just briefly, when the larvae hatch, they're generally not infected with Borrelia because it's not generally transovarially transmitted. So if larvae feed on an infected host, then that's when they could acquire the bacteria that causes Lyme disease. After they molt into the nymph life stage, they may feed on either an uninfected host or an infected host during that time. And since ticks are generalist feeders, they may feed on, during these immature stages, they may feed on any host in the environment. So feeding on a competent reservoir host is how they actually become infected. At the nymph or adult life stage, they can feed on humans, but humans are known as dead-end hosts because the transmission of the pathogen doesn't continue. And adults generally feed on larger mammals such as deer. And while deer are important reproductive hosts for adults, they're not competent hosts for Borrelia, so they won't become infected. So deer are also dead-end hosts for Borrelia. So one of the most important parts of the Borrelia life cycle is in these immature tick stages when the pathogens are transmitted and maintained in this enzootic cycle among ticks and different reservoir hosts. Which is why control efforts generally target either the reproductive hosts, such as white-tailed deer, or reservoir hosts, such as rodents, and mainly Paramyscus leucopus as the primary reservoir host for Borrelia. So in the past 2 decades, there's been considerable efforts in developing tick and reservoir targeted host approaches to reduce the incidence of Lyme disease. And these have had varying degrees of success. So integrated tick management, or ITM, is employing some of those combination of management strategies on an area-wide basis, and it's a vital approach to reduce risk of tick infection, especially when it's in conjunction with other approaches such as reservoir vaccination. But a lot of times these methods have been costly and relatively ineffective over time and large geographic areas. So some previous studies using oral baits have shown a significant reduction in the percent of infected Paramyscus mouse populations, and this was using an OSP-A oral bait. And this, just as a refresher here, this OSP-A is expressed when the bacteria is in the tick, but not when it moves into a host. It's downregulated as it moves into the host. So targeting the ticks during that time is really important. And so another study showed a reduction of nymphal infection prevalence, or NIP, in some areas over time, But this study was using different combination of bait, and they also had some discrepancies in their methodology there. A recent study showed that vaccine deployment with an OSP-A oral bait actually reduced the prevalence and mice that were co-infected with Babesia microti. Um, but in this study, they didn't sample on a yearly basis, so this could actually maybe reflect some normal yearly fluctuations. Um, and we know that Babesia has a higher transmission efficiency when transmitted with Borrelia. So it's really likely that if we remove or reduce Borrelia in these hosts, then Babesia won't be able to be transmitted. So Babesia would, in theory, be lower in these hosts as well. So we found this rodent-targeted vaccine from a company called US Biologics, and we wanted to evaluate its effectiveness at reducing nymphal infection prevalence at some of our participating households. And so this method uses a bacterial antigen expression vehicle, which consists of an OSP-A vectored E. coli system. And so we would use these little lime shield things where we put the bait in there, and they last for about 3 months. And so we deployed them out at households, and this would be a year-long control effort that, as the mouse population um, cycles through, new mice would still get vaccinated, which should decrease infection prevalence in the reservoir hosts and also in the nymphs. So this work was being conducted at those homes that we recruited in our survey in both Pennsylvania and Ohio as part of our Vector Ed Network Training and Evaluation Center. And there's also some notable differences in the types of residential properties between our states in Pennsylvania and Ohio. So in Pennsylvania, we targeted neighborhoods that were adjacent to large city parks, whereas in Ohio, they targeted more rural or ranch type properties. So for the rest of the talk, I'm going to focus just on our work out here in Pennsylvania, which is being led by my PhD student, Emily Beche, with help from several undergrad students in my lab. And as I mentioned, in Pennsylvania, we targeted properties that were in direct contact with large city parks. And so some of these properties actually didn't have fencing or other barriers, and so their yards kind of continuously melded into the park. And these red stars here are some of our recruited households. And as you can see there, they butt right up to the park. And these are a couple of examples of what some of the backyards look like at some of the properties that we visited and kind of where we placed the bait station. So you can see one of those bait stations here in this first picture. And so from our survey, a large number of people reported finding ticks on their property or on people or pets. And so therefore, we went to each of these households and we dragged their backyards for ticks to get a more accurate assessment of ticks on each property. And we found the majority of households did have ticks on their property. Um, and so but one of the issues, so we started this work in 2024, but because of IRB and other necessary approvals, we actually weren't able to start dragging until July, which means that we pretty much missed the nymphal activity, the peak nymphal activity for this year. So we do think that these households probably had higher infection, or excuse me, higher abundance of ticks on their properties. And so we, in the following year, started sampling much earlier in May so that we could account for getting the nymphal peak activity time. So So because we got a late start, we missed that nymphal season, but we still acquired almost 140 nymphs. And of those, about 8% were positive for Borrelia burgdorferi. But we also found anaplasma and some that had Babesia miyamotoi, which causes relapsing fever in Allegheny County, and nymphs from our places out in Westmoreland County were found to be positive with only Borrelia burgdorferi. But again, our sample size wasn't very large, and so in 2025, we increased the number of houses that we visited, and we were able to collect a little over 730 nymphs. And we're actually still waiting on the test results for these. So, because this is a CDC funded project, we were required to send our ticks to a screening lab that met CDC's requirements. So, it's not something that we could do in-house. And so, we're still kind of waiting to get those results back. Um, we also sampled small mammals at some of these properties. So you can see this yellow line here is kind of where we would place traps within that zone there. And we place traps around the vegetation in people's yards and then out into the park border to assess what small mammals were present in the environment and getting into people's yards and possibly bringing ticks and infection closer in contact with people. So we found mainly white-footed mice and chipmunks in both years. And we also were able to sample shrews, which are also important reservoirs for Borrelia and other tick-borne pathogens. And in 2025, when we went to sample, again, because we increased our efforts that year, we collected a lot more individuals, and we also were able to sample a flying squirrel and a gray squirrel. And again, we're still awaiting results, screening results from 2025. But in 2024, we found about 67% of the mice were positive for Borrelia burgdorferi, and actually 100% of our chipmunks were positive. But as you can see, again, these are very small sample sizes. We only had 3 chipmunks, and all of them happened to be positive. We haven't been able to sample or get our shrews screened yet because a lot of the times these shrews are found dead in the traps, and so we have to spend more time to go through and necropsy them in the lab. But we ran into a problem because in 2025 we had deployed about half of our Lime Shield boxes at our properties due to certain delays in product distribution. But then in June, we were informed that US Biologics would no longer be able to supply any product because they were shutting their doors. So we had to figure out a new plan. And since we had already promised our homeowners, our participants, of an intervention, we didn't want to let them down. So we decided to pivot to an evaluation of Bravecto Floralaner product for its effectiveness at reducing ticks on mice and reducing infection prevalence. So our rodent-targeted vaccine project became a rodent-targeted control project. So the use of Floralaner in rodents is a fairly new approach, and it was recently studied by a group in Canada. And their results suggest that it could be effective at reducing the number of immature ticks on Paramiscus mice. As this figure shows the adjusted mouse infection prevalence on the y-axis over the effect of cumulative bait density over time on Paramiscus mouse infection of Borrelia burgdorferi. In a subsequent paper, this group also showed that Floralaner could reduce the density of nymphs and also NIP infection prevalence. And these studies suggest that Floralaner could be a good rodent-targeted oral bait for reducing the density of infected questing nymphs in peri-urban environments. So this was a what we decided to go into for our intervention. And so we had to adjust our study design slightly because the fluorolaner boxes, so more fluorolaner boxes were required to cover the same area that the US Biologics stations would cover. So this is an example of one of our residential properties, which shows about an acre at the site. And so instead of one lime shield station, we're using 12 fluorolaner boxes that are deployed in these three transects, which are shown by the yellow lines. And these are placed 10 meters apart. And each of those contains four boxes. So those are the red dots in there. And so the ticks will be collected along 8 of these 50-meter transects, which are shown by the white lines, so that we can follow the available paths between the bait box transects. And we'll also be um, trapping small mammals at this subset of properties. And we plan to continue this project for 3 additional years to really determine effectiveness over time. To complement our oral targeted control, we're also investigating a topical control intervention using Thermacell tick tubes. And this is the second part of Emily's dissertation work. So she's also leading this study too. And these tubes work by the mice. The cotton is stuffed in these tubes, which kind of look like toilet paper tubes. And the cotton inside is treated with permethrin. And so the mice go in, they take the cotton back to their nests, and so it coats their fur, and then it kills the ticks that are on the mice. So our study setup consisted of 30 100-meter transects. And so this is an example of one of our sites here where we have our, this is mainly done not on properties, but out in our state game land areas. So you can see a path here. And we have the strip of human use area, and then we move about a meter into 10 meters into the forest and set up our transects there. Um, so we had 3 plot types. We had a control, a 2-time deployment of tubes. So we deployed once at the start of the season in April and once at the end in September. And then we also had a 6-time deployment plot where the tubes were placed each month from April to September. So this is an example of what the tubes look like. So they were all flagged and labeled out in the fields. We also dragged for ticks in between those transects. So we dragged for a total of 800 meters, dragging 4 times in between each of those 10-meter transects. And then we also did small mammal trapping on each plot with 20 traps per plot. And so this was done once in June during nymphal peak activity and once in August during the peak larval activity. We're also curious about what other hosts ticks could be feeding on in the environment and also potential predators of small mammals, which may also affect the abundance of these small mammals in the environment. So therefore, we put out audio moth acoustic sensors and also camera traps at our plots so that we could distinguish what other bird species may be present and being hosts for ticks and then other predators as well. So in 2024, we collected some baseline data on nymphal infection prevalence and paramyscus infection prevalence before we deployed any tick tubes. And we found that in our 2 counties, Fayette County and Washington County, there was about 25 to 30% of our nymphs that tested positive for Borrelia burgdorferi, and about 5 to 10% were positive with anaplasma. For our paramyscus mice, the majority were positive for Borrelia burgdorferi, with some of them also positive for anaplasma and Borrelia miyamotoi. So, but again, our sample size was pretty small in this year because it was just trying to get baseline data. And we had about 20 individuals from both locations, mice. So in 2025, this was the first year that we deployed our tick tubes, and we collected relatively similar numbers of questing nymphs from each of our plots. And we found that of the tick tube deployment did have a significant effect on the number of immature ticks that were found on hosts. And where we found that the more often that we deployed the ticks, or excuse me, the tubes into the environment, this helped prevent ticks from feeding on these hosts. And so we're currently testing our nymphs and host samples to be able to determine how the deployment schedule affects infection prevalence. And again, we're planning on continuing this work for an additional 2 years, um, so that we can check and monitor infection prevalence and tick density over time in our different treatment plots. So, as I mentioned, both of our field-based intervention projects are ongoing. And so we are tracking those changes and looking for patterns in tick and host control over time. And from our first summer of tick tube deployment, we did see a reduction in the number of immature ticks on small mammal hosts, with having that more frequent deployment, which showed better results than the less deployment or the control as well. So we're really interested in, continuing this, and especially since we had to pivot over to a new intervention at our households with the floralaner. Uh, we're really interested to see how floralaner could be an effective control method for residents within these different areas. And then also for our tick tubes, we did see that this deployment schedule was effective at reducing the number of ticks on the small mammal hosts, but we're trying to see, will this trend continue over several years? And how does this affect nymphal infection prevalence and also host infection with Borrelia burgdorferi, but also we screen for Anaplasma and Babesia and Borrelia miyamotoi. So stay tuned for hopefully those continuing results in subsequent years. So the last project is I want to talk very briefly about is our lab-based intervention. Um, and I had hoped to be able to provide some information on our lab studies that we're doing to evaluate the effectiveness of some new tick control or tick repellents. However, the university has prohibited me from speaking further about this. So um, I'm really hoping that we can have some results and publish them maybe by the end of the year. So kind of stay tuned for that also. I think there's some really interesting stuff we're going through, and I apologize for not being able to talk further about that. So this research really couldn't have been completed without the hard work, very long hours, and great dedication of our summer students, both graduate students, and we had a number of undergraduate students as well that um, some of them were volunteer, and so they, really helped move these projects forward. So I'd like to acknowledge those students as well as my PhD student, Emily, for all of her hard work on this project, and my colleagues from the Vector Ed Network, and also thanks to the CDC for funding this work. And with that, I'm happy to take any questions.
Thank you so much for your presentation, Dr. Tufts. I'm going to launch the post-webinar quiz question while we take questions. So if anyone has questions, please put them in the chat or raise your hand. We've
Should I
got
stop sharing my
plenty of time.
slides?
Up to you.
I think I'm going to stop so that I can actually see
Okay.
the chat.
There's a big difference in these answers this time for the post question. Lots of people getting it right. Any question?
Sorry if I sped through that. I it went a little faster than I expected.
Is there anything you want to expand on? Okay, we've got a question. Thanks for sharing the map of states with and without KAPs. Is there a place, maybe one of your publications, where we can see the list of all the studies?
Yeah, so in that paper that is from my student, so you can find it using participatory epidemiology by Beche et al. In 2025. In the One Health Journal, we do list a lot of the other places that have CAP studies done. Do you know if the results of your study with them?
I'll read it out.
Yeah, and so I see this question from Evelyn. Do you know if the results of your study with the homeowners influenced any of them towards wanting to use these tick control methods on their own? So one of the issues with the US biologic study is that it wasn't available for homeowners to get on their own, which is another reason why we kind of wanted to switch to something different. So the floor laner, they can actually um, purchase that. You know, you can get it for your dog or your cat from your vet. And so it would be a method that people could use on their own. And what we're planning on doing is after the study, doing another survey of the participants, especially the ones that we had the intervention at their property to see did their perceptions change, did their knowledge change, would they continue the intervention past the study time, things like that. So, but I do think that given the information that we've given them already, a lot of people have been interested. It's funny, neighbors talk to each other too. And so from 2024 to 2025, we did have adjoining properties that when they saw us out at the property came and said, "Oh, you're the tick research group. Can you include my property in this study too?" So we do think that it is having an impact, especially within neighborhoods. Um, and Emily did post a link to our paper. So thank you for that, Emily. Um, Emily, did you change your personal protection kit strategy in the past 12 months?
Hello, everyone. Um, I don't know that I changed it much. Going into this lab, I was very cautious of everything, but I spray all of my clothes with permethrin throughout the season, and I put a bunch of different repellents on when I'm out in the field.
And I do want to mention that they do wear more protection. A lot of those pictures were uh, ones that I asked of them working. And so um, it's not all the time clear, but they do have protective suits that are coated. They do put their pants in their socks, and they do use a lot of personal protection while in the field too, which may not be apparent from those pictures. So,
Great. Does anybody have any more questions? We have lots of time. All students treat their clothes with permethrin.
yeah, we do provide DEET and permethrin for the students that are working. We provide those they're like the full body painter cotton suits that we spray with permethrin. So we do make sure that everyone is safe when doing this research.
Do you also use repellents while tick dragging? And if so, do you find that having them on makes it more difficult to attract or collect enough ticks?
Yeah, that's a good point. So when we are doing tick drags, we don't use the spray on. We only have the treated clothing because we don't want it to potentially get on our hands and on the drag cloths and affect our ability to collect ticks while in the field. So yes, that's a good point. We don't do the spray when we're doing the tick drags.
Yeah, good question. Glad to hear about all the interest in this. Okay, last minute questions here. Think
Yeah,
that.
anything else? I'm happy to answer. Yeah, again, I was really hoping to be able to talk more about our uh, lab-based intervention, but was told kind of last minute you're
Yeah,
not allowed to. So
that's understandable.
yeah.
Well, looking forward to hearing more about that and the results and everything. Um, Thanks again for being here.